Now that we've gone through the light reactions and we've converted light energy into chemical energy in the form of ATP and the NADPH in the stroma of the chloroplast, we are now ready to build some sugars in the second half of photosynthesis. Here's where the Calvin cycle comes in. The Calvin cycle has a lot of similarities to the citric acid cycle, meaning that it will regenerate the starting material that it starts with so that the whole process can go full circle or be cyclical. So what we're going to do next is we're going to take a look at the Calvin cycle. We're going to see how the Calvin cycle takes simple molecules of carbon dioxide, and with the energy from ATP and the reducing power of NADPH, it builds sugar molecules. All of this is going to take place using enzymes that are found in the stroma of the chloroplast. Okay, so if we think about this for a minute, the sugar that's made by the Calvin cycle is not actually glucose. The sugar that's made by the Calvin cycle is a simpler sugar. It's only 3 carbons, and this sugar is called glyceraldehyde 3-phosphate, or G3P. I'm hoping that somewhere in the back of your head that rings a bell. And hopefully, maybe you remember that G3P is actually the sugar that's produced right in the middle of glycolysis and cellular respiration when the original 6-carbon glucose molecule gets split right down the middle. And during that split in the middle of glycolysis, way before pyruvate is made, those 2 3-carbon molecules are molecules of glyceraldehyde 3-phosphate, or G3P. So while photosynthesis doesn't directly make glucose, it does make G3P. And if you take 2 G3Ps together, you can kind of go backwards in glycolysis and you can make glucose very easily. And then glucose can be used by the plant cells as a source of fuel, or to build starch, or to build cellulose, right? We can do lots of things with those glucose molecules. Now, if we're going to build G3P, a 3-carbon sugar, how many carbons do we need? We need 3. Where do we get carbons to build these organic molecules? We get the carbons from molecules of carbon dioxide, CO2. So if we want to build a 3-carbon G3P, how many carbon dioxide molecules are we going to need to build one G3P? Hopefully you said, we're going to need 3 carbon dioxide molecules because each carbon dioxide molecule only has 1 carbon. So if you need 3 to make G3P, 3 carbons, that means 3 carbon dioxide molecules. So when we're looking at the Calvin cycle in the next few slides, we're going to be looking at the full cycle. And really when we're looking at this full cycle, we're looking at it taking place 3 times at the same time, right? So we're going to be taking 3 carbon dioxide molecules initially to make 1 G3P, using 3 turns of this cycle. Now, the Calvin cycle has 3 main chunks, and these are the 3 main chunks that I need you to know. Again, we're going to go through the individual chemical reactions, most of them, for the Calvin cycle. I don't need you to memorize all of those. I just need you to know the main 3 processes that take place in the Calvin cycle. So, first phase is called carbon fixation. This is catalyzed by an enzyme that's called Rubisco. Rubisco is thought to be one of the most abundant proteins on the planet because every organism that performs photosynthesis will have this enzyme that helps to fix carbon from carbon dioxide into an existing carbon skeleton. The existing carbon skeleton that the Calvin cycle and Rubisco use is a molecule that's called RUBP. RUBP is the carbon dioxide acceptor. Interestingly enough, RUBP has 5 carbons. Carbon dioxide has 1 carbon. So the process of carbon fixation, this first phase of the Calvin cycle, is to take that 5-carbon RUBP and attach the 1 carbon from the carbon dioxide to it. Step 1. Step 2 is reduction. You have no idea what happens during this part, but you know that it's called reduction. Take a guess on what you think will happen during the reduction phase of the Calvin cycle. hopefully you said that something is going to be reduced, meaning something is going to accept electrons. So yes, as we go through the process of building the RUBP, we're going to be using electrons that are coming in from the light reactions that are being carried by the NADPH. We're going to take those electrons and build them into the sugar molecules that we are constructing in the Calvin cycle. And really at the end of reduction here, we've already made our G3P. So the last stage, the third stage of the Calvin cycle, is really to regenerate the RUBP that we used at the very beginning of the cycle, so that again, the whole process is cyclical. All right, so let's go ahead and then take a look at this Calvin cycle process. How do we take carbon dioxide, convert it into that G3P, using ATP and the reducing power of those electrons from NADPH to make the sugars? And then how do we make it full circle so that we can regenerate those molecules, the RUBP molecules that we need at the beginning of the Calvin cycle, to take another spin around the block? All right, so here's phase one, carbon fixation. Here's exactly what I was talking about when I said we take an existing 5-carbon skeleton, RUBP, and we fix a carbon to it from the carbon dioxide. This is the first step. So right here, you see here's our 5-carbon, 1, 2, 3, 4, 5-carbon RUBP, and here are carbon dioxide molecules. We're going to take 1 carbon dioxide, 1 RUBP, and stitch them together to create this 6-carbon molecule. And the 6-carbon molecule is so unstable that as soon as it's made, it's actually broken in half. Broken in half to make 2 3-carbon molecules. These molecules are called 3-phosphoglycerate. Now, remember that on the previous slide I said, well, to make 1 G3P, we need to use 3 carbon dioxides, and we're going to go around the circle 3 times. So really what you're looking at is everything in triplicate. So we've got little number 3 here. That's telling you we're going to use 3 RUBPs. We're going to use 3 carbon dioxides. So 1 carbon dioxide per RUBP to create 3 of these 6-carbon molecules. And if you take these 3 6-carbon molecules and split them all in half, you end up with 6 3-carbon molecules instead. So now we've incorporated the carbon from the carbon dioxide into a carbon skeleton. So from here, we have to remodel this molecule to make our G3P. And this is the second stage of the Calvin cycle, which we call the reduction phase. So here, we're going to use a lot of ATP to reorganize the molecules, using the phosphates there from the ATPs. We're also going to use a lot of those electrons from the NADPHs. So this is where the reduction takes place. We're going to add the electrons to these molecules that we're using as intermediates to eventually make the G3P. And so at the end of the reduction phase, we have made now 6 G3P molecules. That seems like a lot, right? Really, really high throughput. There's a catch though, right? Actually, only 1 of these G3P molecules is a product. The rest of the G3Ps have to stay in the cycle so that we can complete the cycle, right? We've got to fill in this gap right here. So let's do some math for a second, right? If we have to fill in this phase, the regeneration phase, right? We have to remake 3 5-carbon molecules of RUBP. So do the math with me. How many actual carbons are there here in these 3 molecules? Well, 3 times 5 is 15. So we need a total of 15 carbons to remake those 3 RUBPs. How many carbons do we have here at the end of the reduction phase? Well, we've got 6 3-carbon G3Ps, so 6 times 3 is 18. We've got 18 carbons here. We need to keep 15 of them so that we can remake these 3 RUBPs. So 18 minus 15 is only 3. So really, again, only 1 3-carbon G3P is the output. Honestly, it also kind of makes sense because if you think about it, right, you can't create, can't destroy matter, so whatever matter goes into this cycle is the same matter has to come out, right? So how many original carbons do we put in? We only put in 3 carbons from 3 individual CO2 molecules. So only 3 carbons, aka 1 G3P, can come out. Okay, so we've made our product. Now we need to regenerate the RUBP. So here we're going to take the remaining 5 G3Ps, and through the use of some energy, ATP, we're going to reorganize to take the 5 3-carbon molecules and instead now make 3 5-carbon molecules. We have finished where we started. These 3 5-carbon RUBPs now can be used again for the first phase to fix 3 more carbon dioxide molecules. All right, and so this figure kind of gives you a nice summary, gives you the 3 phases: carbon fixation, reduction, and regeneration of the RUBP. It also might help you sort of get a handle on how many molecules and how many carbons in those molecules you have for each of those phases. So at the end of carbon fixation, you end up with 6 3-carbon molecules. One of those gets turned into the G3P. And then the remaining 5 3-carbon molecules get remodeled to create 3 5-carbon molecules instead, so that you can again go through carbon fixation. Okay, on Canvas, I've got this BioFlix animation, which walks you through photosynthesis, the light reactions, and the Calvin cycle. If, again, you can narrate this particular BioFlix animation, and it doesn't go through all the details, but if you can narrate it, if you can explain what's going on there, then you have a pretty good grasp of the process as deep as I want you to go in it. So make sure you review that at least a handful of times. And again, that's in Canvas. All right, so the last slide is a little bit of a review for photosynthesis, right? So we talked about photosynthesis as this process where sunlight really gets converted into chemical energy and is stored in these organic compounds. The initial sugar that's made, that G3P, can then be used for a variety of different processes in the cell. That G3P might be used to make glucose, which could supply energy to the mitochondria, for example, or it can be used to build carbon skeletons. It can be used also to make a variety of different molecules, including things like cellulose. It can also be stored as excess energy in the form of starch. Remember that plants store this excess energy in the form of starch in a variety of different structures: roots, tubers, seeds, and then fruit. And then it's really important to remember that in addition to making food, photosynthesis is a process that also makes oxygen, and that oxygen gets released to the atmosphere. And both the oxygen and the sugars are the reactants for cell respiration that all cells that do aerobic respiration are going to use to make ATP to drive all other cellular processes. So even though we've been focusing on plants as these organisms that perform photosynthesis, don't forget that they're making these sugar molecules via photosynthesis so they can send them into cellular respiration in their mitochondria to make energy for the cells, or to convert energy into a usable form for the cells, so the cells can go about paying for their cellular work to keep the plant cells and the plant itself alive.